I guess I'm first here. So I'm a basic scientist. I have not had previous interests in working with industry or doing anything commercial, so this has been like an eye-opening experience for me. So what I'm going to do is I've got just a handful of science slides which sort of explain the project very, very generally, just to give you an idea of where I'm coming from. But understand that, again, I'm a basic scientist. I look at basic questions that are really, I didn't think would be applicable in a commercial sense. Okay, so what we're looking at is a problem in cancer, and I think we all realize that cancer is a really problem both the US and worldwide. And what's really the problem in cancer is metastatic disease. That is when the cancer cells leave the primary site of the organ and migrate to other parts of the body. Most solid tumors that are located in one place can be treated surgically, so that's really not a life-threatening disease. It can be in some cases, but when the tumor starts to spread, it goes literally to hundreds of different locales in the body, and that's not something that can be treated surgically. I think we all know that chemotherapy and radiation therapy are the current models that are used. We also know the side effects of chemotherapy. They frequently kill the patient before the tumor does, and radiation may or may not be effective. Again, depending when it's disseminated, it's a real problem. So I'm an immunologist, and we have known for probably 100 years that the immune system is capable of actually killing tumor cells. And the problem has been, how do you get the tumor to actually kill the tumor? How do you get the immune system to kill the tumor? The cells of the immune system that are really important are cells called T lymphocytes, or T cells. I'm just showing in blue. We know they can do it, but it's a matter of manipulating the system to get that to happen. So we call, and what's come to known in really the last 20 or 30 years is the phenomenon of immunotherapy, where we're trying to actually activate patients' own immune systems to respond to their tumors. Again, I'm a basic scientist, so we did all of our work in animals. We never actually did much, we never did any work with patients, and we're trying to understand the mechanisms, how you get those T cells to migrate to the tumor, how you get them, once they're at the tumor, how do they interact with the tumor. So in many cases, the immune system can respond to tumors, but it doesn't happen spontaneously, and this is where the project comes in. We know that once T cells are activated, because they have to go from sort of a resting state to an activated state in order to respond to the tumor, and once that happens, they also begin to express receptors which will kill themselves. And the idea for that is that if you have a viral infection, you activate your T cells to kill the virally infected cells, but then you kill those cells, and then you don't want those T cells anymore. You want those to die so the next set of cells can expand and kill the next viral infection. And of course, in the case of tumor, you don't want that to happen. You want to keep those T cells alive. So the T cells actually suicide after they become activated. And one of the molecules, again through basic science that's been discovered on T cells that causes them to die is something called programmed death 1, very appropriate name. And what happens is that the trigger to kill the T cell, so the T cell has this programmed death 1, PD1, and the tumor has that ligand that's going to bind to the PD1 and kill the T cell. So the tumor is very well adapted to express this PDL1 molecule. It cozies up to that T cell, which is trying to kill the tumor, and bingo, the tumor instead kills the T cell, and the whole immune system that we've tried to activate is useless, and the cancer progresses. So just showing you schematically, if you have a T cell, it's going to have that PD1. The little red thing is supposed to be the PDL1. It's on the tumors, which are shown in red at the bottom, and that interaction just kills the T cell. So one of the concepts that we stumbled upon literally was that if you prevent this interaction between PD1 and PDL1, you can actually get the immune system to remain activated. And now there have been recent clinical trials, and this is really where the commercialization comes in. People have now developed antibody molecules, proteins to PDL1 and PD1. They've been tested in clinical trials, and to a certain extent they work, and some patients are actually cured. But antibodies aren't completely effective. They only really help a subset of patients. And so we need to develop better therapies. And so in order to block PD-L1 and PD-1, we obviously want to do this so we can maintain those activated T cells to keep killing the tumor. And so what happened in our case was that under MII support, and before we had it, we literally stumbled upon an alternative mechanism to block this PD-L1, PD-1 interaction. And what we're trying to do now is actually develop that approach commercially so it could be used therapeutically in the clinic, and I think that has obvious commercial advantages. And just my last slide here on data, just to show you that it actually works. If we take our T cells, in this case we're using human melanoma cells that express PD-L1, which is the little blippies. Oh, I'm not going to get this. All right. Well, the cells are the red, and then the PD-L1 is supposed to be the little red things hanging out from there. We then come in with our therapeutic, which is something called CD80 Fc. I don't really need to explain what that is, or antibodies. And we ask, which of these agents are more effective? Antibodies being what's currently being used, and that's partially effective. And so if we just do a scale here of T cell function, how good are those T cells? The higher the number, the better they are. If we just have those T cells in the absence of tumor, they work really well. They're very activated. If you add the tumor cells then that express PD-L1, your T cell function just goes down because those T cells get killed. If you then come in with antibodies, and these are two different antibodies, you partially restore reactivity, but not completely. If you come in with antibodies to the other molecule, you again partially restore. But if you come in with our reagent that we're developing, it's much better than either the antibodies to either one. And so, and just as a control, an irrelevant protein isn't going to do anything. So that's basically what the project is, and we're really continuing to develop that. So in terms of what the MII and also the Catalyst program has done for us, so we never really thought that what we were doing had any commercial impact, because we were looking at basic mechanisms. How do you block this suppressive mechanism? And when I first, I guess, got in touch with her, when David, I'm not sure if you got in touch with me or I got in touch with you. I can't even remember how we got connected, but it just sort of set the wheels spinning on this really could have some potential commercial development. And I really hadn't thought about that before. Dave also put me in contact with an entrepreneur, and himself, he's a UMBC facilitator, the minor right there, and they really helped me to present the project in a commercial sense. I'm used to writing NIH grant applications. It's a totally different ball game in the way you write the application for a grant as opposed to when you write something for commercialization. They were really instrumental in guiding me into how to present it, what the cosmetics looked like. And then, of course, MII provided funding to generate the additional data that we needed to pursue. Now, we actually didn't go to the phase 2 and phase 3 of MII because a company picked this up, and the project is now being funded by MedImmune. And there was some discussion there about whether we should continue this, try to do a startup, or whether I should go with MedImmune, but the MedImmune offer was literally too good to turn down, so we went that way. The other thing that happened to me through the MII program was that the collaboration that Dave set me up with, the entrepreneur, led to another project. Also a project, an immunotherapy of cancer project, but a completely different approach to how to facilitate the immune system. And that program then is now being, or that project is now being funded by the Catalyst program, because it sparked an idea, again, put me in contact with someone who I could actually work with. Because again, I hadn't had the contacts with people who were industry savvy in order to know how to do these things. And so the Catalyst funding, which we literally just started, I don't know exactly where that's going to go, but I hope we can get through that program enough preliminary data to now submit another MII proposal so that we can keep going again along the way. So writing these programs, I must say, writing these applications and working with people who are interested in commercialization has really opened my eyes to a whole new set of ideas, and that also sparked us to be able to submit a UMBC-UMB joint collaborative grant, which is now, I would say, about 80% basic science and about 20% commercialization development. So this has really been an eye-opening experience for me, and it's kind of shifted the whole way that I look at supporting my lab. That's it.